Hey guys, so today we're going to be finishing up our discussion of cellular respiration by talking about what's happening at the electron transport chain. So when we left off, all of the high energy electrons from our original glucose have been moved over, reduced into NADH and FADH2. So all throughout glycolysis, the transition step, and the Krebs cycle, we've been taking electrons from glucose and putting those high energy electrons into the electron carriers, NAD and FAD, and they're going to now shuttle those electrons over to the electron transport chain. So the electron transport chain is located in the cristae membrane of the mitochondria. So remember, mitochondria are double membrane bound. They have an inner cristae membrane and then they have an outer membrane. So when we're talking about the electron transport chain, we're talking about all of these colorful jelly beans that are here inside of this inner cristae membrane. Each one of these colorful jelly beans is a protein that's going to move or shuttle the electron just slightly lower in energy, establishing a proton gradient. This is just like the electron transport chain of photosystem 2 in photosynthesis. So those two electron transport chains are very, very similar in their structure and function. So let's look. We have our NADs and our FADH2s, and we also have our outer mitochondria membrane, and then inside of that we have the inner membrane. So in this inner cristae membrane, we have this electron transport chain. And so I'm going to put the electron transport chain in here as some proteins embedded into this area. So the number of proteins that are in there is not terribly important to the study that we're doing of this, but this is an accurate picture that's on here with each of the coenzymes and the cytochromes that are going to be working to pass the electron along. Memorizing these proteins is something that you might do when you're taking AP Bio or if you retake this again in college. So what's going to happen is our NADs and FADs are each going to travel over to the electron transport chain. And they are going to drop off an electron. So this is NADH here. It becomes NAD plus. And now this NAD plus is going to go back to go pick up more electrons. So NADH at this point has done its job. It has dropped off its electron at the electron transport chain. Now what happens here is a very fantastic game of keep away. The electron is going to be kept inside of the proteins, trapped inside of the proteins, and it's going to travel down slowly, protein to protein, losing energy as it goes. And so again, this is a series of redox reactions. It gets picked up here because this protein likes it, and then no, this protein likes it, and so it pulls the electron over. No, this protein likes it, so it pulls the electron over. So each one of them pulls this electron down, down, down to a safer, happier location for the electron. Each step it takes, it's taking one more step towards happiness, okay? In the meantime, there are also protons. Remember the proton gradient of photosynthesis? This NADH, remember the H can split. And so as the electron gets traveled through the electron transport chain, the protons are moved over and establish a proton gradient over here. So we're getting this high concentration of protons again between the outer membrane and the inner cristae membrane. We're getting this high concentration of protons in there. And then the rest of this story actually looks kind of similar to what we see again in photosynthesis. At the end of the electron transport chain, we're going to have the final electron acceptor of cellular respiration. This is a big time test question. This is always a test question. What is the final electron acceptor of cellular respiration? And the answer is oxygen. This is a weird little symbol we write here, a 1/2 O2, which is just a fancy way of saying oxygen, right? If you have 1/2 of a pair of shoes, you have one shoe, right? If you have 1/2 of a pair of oxygens, you have one oxygen. So we have a 1/2 of an O2 that's going to combine with the electron. So we have our 1/2 O2 combining with an electron and combining with some protons that have been allowed through. And so we end up with this H2O, or water, right? So we've got our little drop of water being produced here in the body. The most important part of all of this is what's happening meanwhile. Meanwhile, all of this is happening. This molecule, which you might recognize as ATP synthase, is using the drive of the electrons, of the protons wanting to come through in order to make ATP. And so this, right here, this is why we do cellular respiration. This is why we need cellular respiration. This process kicks out about 28 ATP. 28. In all of the rest of this time, we made 4 ATP. But the NADHs and the FADH2s, coupled with the electron transport chain, are going to kick out 28 ATP per glucose. That's enough energy for you to grow so big and for you to think so many thoughts and for your body to grow to be so many cells. They need a lot of energy. And so aerobic cellular respiration is what allows for that. The part of this that is aerobic is this little guy right here. Aerobic cellular respiration with oxygen. Okay, we are with oxygen. And so we call this chemiosmosis. Osmosis is the movement of water. This is the chemical movement of water. We end up moving water across the membrane, but in a chemical or broken down way. Chemiosmosis, aerobic respiration, and the other term you're going to hear is oxidative phosphorylation. We need oxygen as our final electron acceptor in order to do this ATP phosphorylation. So this is just a breakdown of the different terms that we're using. I like this one just simply for that. So we see the electron transport chain right here. The electron transport chain is this movement in pink here of the electron that establishes the proton gradient. And then we have chemiosmosis. Chemiosmosis is happening here, the movement of the protons across forming water. We call this whole process, everything over here, we call oxidative phosphorylation. So that's a really nice, I think, breakdown of the terminology that we're using. So this is just one more way to explain it. Oxidative phosphorylation, which is this part, right? Adding the phosphate onto ATP, adding the electron onto oxygen. Via chemiosmosis, so we are doing this movement of hydrogens across at the electron transport chain, because the electron transport chain is creating the environment that's going to allow for the chemiosmosis that does the oxidative phosphorylation. Phew, that's a lot. So this is using the energy that is created when a proton crosses along its concentration gradient to do something that requires energy. So we call this part where the proton goes across exergonic, because energy is exiting, energy is being given off. And we call this part where the ADP is becoming ATP and organic, because that uses or stores energy. And so that's just another way to describe it, yet more words to say the same thing. This is a more complicated look at what ATP synthase looks like, mostly just to drive home the idea that it takes 3 or 4 hydrogens to go through the ATP synthase molecule for each ATP that is made. And so whenever I draw it, because I'm limited, my drawing does 1 hydrogen making 1 ATP, because I don't ever have enough space to show how many there are, but it actually takes multiple of these protons coming through before any ATP is able to be made. Okay, so when I see you guys next, we will draw this whole thing all together, start to finish, start with the glucose, end with water, make 32 ATP. We'll draw it all out. We'll make sure we have it nice and centered in our minds. So I will see you guys in our next class for that.